Hello, welcome to your own channel, Curious Corners in Biology, where we delve into the scientific inquiry of the subject and extract curious questions in this subject. So, this is a channel which makes the subject of biology more interesting in a question answer manner. This is for all curious learners, irrespective of any age, young or old, but who has curiosity in the subject. Of course, this content will appeal to you. So today we are going to discuss an interesting topic about mitosis. Mitosis is a form of cell division which is observed in eukaryotic cell. And there is meiosis as well, which is more complex form of cell division observed in eukaryotic cells. But a question which arises is that how this cell division mitosis might have evolved in eukaryotic cells. To answer this, we know that prokaryotic cells might have evolved into eukaryotic cells. So that means the cell division in prokaryotic cells might have evolved into mitosis in eukaryotic cell. What do we know about cell division in prokaryotic cell? How does it occur? Binary fission. So binary fission is an amitotic type of cell division, meaning to say that it is non-sequential, it does not involve sequence of steps, and it is not complex. It is very rapid and easy. Cell simply divides into two cells by formation of groove along the cell membrane. The nucleus, that means the nucleoid, the genetic material, is distributed evenly into the resulting daughter bacterial cells. And in mitosis, we observe sequential steps like prophase, metaphase, anaphase, telophase, and then finally cytoplasm divides. So there is karyokinesis followed by cytokinesis. So naturally, from prokaryotes, certain eukaryotes might have evolved, and there might be some intermediate stages of cell division which would reflect the evolution of binary fission into mitosis. Okay, so let us understand this. I'm sharing the screen and you might see whiteboard. So if we are thinking about a bacterial cell, how does binary fission in a bacterial cell takes place? So say this is a bacterial cell, okay? And let's say that this is the nucleoid or the genetic material in a bacterial cell. Now when this bacterial cell progresses into binary fission, a groove develops like this, and the genetic material attaches itself along the plasma membrane or the cell membrane in a way that it is ready to be distributed equally. And when this progresses further, the cell simply divides into two cells. The cell simply divides into two cells like that. This is how binary fission progresses. And this is seen in bacteria. So let's say that this is binary fission in bacteria. All right. Now, eukaryotic cells which have evolved from prokaryotic cells and which are very near to prokaryotic cells are dinoflagellates, diatoms, yeast. Yeast is a fungi. So, what happens in dinoflagellate? Suppose this is a cell of dinoflagellate. Now, in dinoflagellate, nuclear membrane remains intact during mitosis. Spindle fibers form in cytoplasm and they get into the nuclear membrane and drive the chromosome movement. So here, the nuclear membrane is intact. Okay. You have chromosomes like that within the nuclear membrane, and you have spindle fibers like that radiating from cytoplasm and running into the nuclear membrane. Okay, so this cell eventually divides like that. The nuclear membrane shows formation of a groove. The nuclear membrane shows formation of a groove, and finally the cell divides into two. I'm not drawing the intricate other details of the diagram. Now you can clearly understand that chromosomes are distributed and the cell divides into two. This is what occurs in dinoflagellates. So this is a version of mitosis which is called as endomitosis. This is called endomitosis. In diatoms and yeast, what happens is the same version of endomitosis takes place, but the spindle which is formed is also intranuclear. In dinoflagellates, the spindle which is formed is running from cytoplasm into the nuclear membrane, but in diatoms and in yeast, the nuclear membrane inside of it shows formation of spindle. So in this manner, if we have chromosomes, then spindle is formed within the nucleus. Spindle is formed within the nucleus like that. So this is called intranuclear spindle. And finally the cell divides into two. Okay. We are not discussing the entire mechanism over here, but just to get an idea of how this variant of mitosis progresses. Okay. So ultimately, the cell divides into two. Does not exactly progresses the way I have shown, but this is just an assumption. Finally, we will have two cells like that. So this is the endomitosis which occurs in diatoms and yeast. So broadly, what do we get from this concept? From this concept, we understand that binary fission might have evolved into different variants of endomitosis, and then endomitosis would have evolved into mitosis, what we see in eukaryotic cell. And to support this sort of evolution, there is a specific protein, FTSZ protein, which is homologous. That means structurally similar to the tubulin protein, which organizes or polymerizes the spindle fibers or the microtubules of eukaryotic cell. So this FTSZ prokaryotic protein is structurally similar to the tubulin protein responsible for organizing microtubules or spindle fibers of eukaryotic cell. So this is how we understand that how binary fission might have evolved into mitosis. So hope you did find this discussion interesting, and further on we are going to discuss questions along the same lines. If you are interested in such content, please subscribe to my channel, and in the channel description or in the video description, you will find a link of my book Curious Corners in Biology, which is a dungeon of such questions, and it is available at Amazon Kindle at a very affordable price. You can obviously purchase the book. I am sure that you will not get disappointed reading the book. Okay. Thank you very much. See you soon next time. Thank you.